Thanks for the introduction, Elena. I will share my screen and welcome everybody. Good morning, good afternoon, or wherever you're located. Thank you for joining me today. The webinar today is dedicated to the demanding process of drug discovery and development and how to accelerate it with single-cell data tools. So after an introduction into the topic by myself, Denise will walk you through the software in a live demo. And any question that you might have during the webinar, you can enter via the question and answer function of the Zoom window, and we will collect the questions, and there will be a question and answer session later on. So let's start. Few words ahead regarding Singeron Biotechnologies. We are an international company with a headquarter in Cologne in Germany. And next to our R&D and engineering department, also the European Service Lab is located here. We Singeronis are totally addicted to single-cell analysis. We develop applications, kits, instruments all along the workflow, starting from preservation and dissociation of fresh tissue for high-quality single-cell solutions, over proprietary chip-based analysis with or without instrument and automation, to bioinformatic tools to get the most out of our data. So if you have any samples or project ideas, but no clue, and just heard that there might be some between you who are new to the topic, so if you have no clue how to start single-cell analysis on your project, just drop us a line, and same, of course, for any other participants and any other questions you might have. Because today, we skip the wet lab and sequencing part and work exclusively in silico. So our starting material is single-cell data, no matter which data format or which platform or technology it comes from. Okay? Drug discovery. Drug discovery and development. That's, yeah, all of us appreciate the proved medication in times of needs, but few are bold enough to take on the challenges of new development. So it starts with understanding the disease, or at least relevant pathways, and identifying suitable targets. In human bodies, organs, and even single cells, the molecular connections are complex, and it is often impossible to decipher all details of the disease mechanism. So the need to finally reduce everything to a handy model for high throughput screening doesn't make it better. So, well, luckily, we are not alone in this. Out there are so many researchers, and they're usually happy to share their results, at least if they're in academia. Quite likely, there is supporting insights, papers, or even public data. But even if we get hold of it, each paper has a distinct data set and often customized bioinformatic data processing. So how to combine and how to correlate the public data with my own data. When screening for suitable compounds finally was successful, lead optimization and the preclinical tests on cellular and animal models add complexity again. So the more complex the model, the more costly the testing gets. So better to know early than lose a candidate in a later phase due to unexpected side effects. At this phase, single-cell data could serve as a glimpse into the crystal ball, as it makes subtle effects in specific cell populations visible that might get lost in bulk analysis. If we would have easy access to consolidated datasets. Finally, in clinical trials, yeah, it's delicate in its own way. Not for nothing, there are legions of service providers dedicated to recruiting subjects and conducting and documenting the tests. It is an art to collect a cohort of suitable persons with the appropriate characteristics at the same time. For some of the indications, the limiting factor is not even costs, but the low number of suitable patients. Wouldn't it be helpful to complement the patient cohort by adding data from in silico sources? But you might have already suspected it. We have prepared something. A pair that complements each other. CeleLens, a tool to analyze and visualize single-cell data, and SynEcoSys, a curated single-cell database. So our CeleLens platform is perfect for all of us brilliant researchers who, unfortunately, have no clue about bioinformatics, but still insist to analyze our data ourselves. Would never give it to someone else. Whatever technology you use, whatever data format, whatever scientific question you want to answer, CeleLens is your go-to software. The single-cell RNA database SynEcoSys is packed full with public single-cell datasets, but not just collected. The data is manually annotated in a standardized way. So SynEcoSys is the magic wand that turns the vast amounts of crude data into a usable resource. A few more words on SynEcoSys. It includes human single-cell data, mostly clinical and often including additional clinical information. By the way, clinical means diseased, but also treated with different drugs. Very useful for monitoring side effects and also for repurposing attempts. Marine datasets obviously serve as extension for preclinical tests. And lately, we also added some non-human primate data, especially interesting for neurological fields. This is what SynEcoSys does. On the left, we see 3 distinct datasets, same topic, but different focus of research and individual naming of cell types. We would need to look at it one by one. On the right, SynEcoSys consolidated the datasets as one database with consistent annotation of cell types. In this comprehensive database, we can now search for whatever we like. Let's say we identified one specific target. It seems to be crucial in the pathway we look at. So now we want to know how the expression of the gene is affected by medication to check for possible drug repurposing, for example. This is what it would look like in SynEcoSys. We see expression of our gene in various cell types in relation to treatment with 14 different drugs. In one view, we get the answer to our question from 14 distinct publications and 14 individual datasets. And it's not only drugs. You can browse by response or mutation, by disease subtype or model that was used. So the analysis is possible on gene level, but also on cell or tissue level. And in addition, you can even narrow down the data source from all available data to specific publications, maybe for your topic of interest, whatever you like. Coming back to drug discovery and development, we see SynEcoSys useful at several stages. In the early R&D, we can dig into the data for novel targets or verify the expression of already identified targets. The data can also be used to support drug repurposing, as mentioned, or expand the use to additional indications. So during preclinical tests, we can run in silico toxicity tests, much cheaper, and monitor side effects. But SynEcoSys can also predict effects of combined therapies. To save time and money in clinical phase, we could better select suitable patients and also extend the database in case of small cohorts. Does this sound too theoretical for you? Let me show you an application. For example, mining a novel target. Let's say we are looking for a drug target in colorectal cancer treatment. So we search SynEcoSys and find KLKL gene family that pops up with a high expression level in colorectal tumor tissue. We look at it more in detail using two colorectal cancer specific publications, or rather the corresponding datasets. This is the SynEcoSys analysis results of dataset number 1. It confirms tumor-specific expression of KLKL6 and KLKL10. Quite obvious. Same for dataset number 2. So it seems KLKL gene family might be an interesting drug target in the context of colorectal cancer. We have quite some partners who successfully work with these tools. So Beyondus is one of them. Beyondus is a Belgian biopharmaceutical company focusing on intractable cancers and autoimmune diseases. They have a pipeline of 7 compounds targeting oncological indications, including 3 candidates in phase 1 trials, so first in human studies. Beyondus is already successfully using SynEcoSys since a while and lately also added CeleLens to their toolbox. Right. By now, you must be curious to see the tools in action. So I hand over to my bioinformatic colleague, Denise, and she will show you how it works.
Okay, thank you, Regina, for your nice presentation and introducing me. So today I will basically walk through the SynEcoSys, the single-cell RNA database, and then the CeleLens Cloud Analysis platform on a live demonstration. So I don't have a presentation, but we will go over both platforms live together. Let me share my screen. Starting with SynEcoSys database. So it's a collection of single-cell RNA and single nuclear RNA datasets and different publications. And this is the homepage of the database itself. So this big welcome page actually has everything you need. So starting from the top, we have like a small, like a search bar here for a quick search, either by genes or cell types or different studies. And with the search bar, you can just search for anything you're interested in. For search function, we also have this browse by tissue atlas. I think I will show you this one first. So if you're interested seeing different parts of and organs of different species, you can just browse by the tissue or cell atlas. So here in the database, we have datasets coming from human, mouse, and some non-human primates. And here with this nice representation, you can see different organs and their subsequent tissues and cell markers on top of a cell atlas here on a representation on top of the bodies. So here, if you're interested in one of the organ systems, you can just go and switch between them. And then let's say you would like to search something in lung tissue or the organ itself. And then you would like to maybe see some cell type markers of one specific cell type coming from lung. So you can just click on the cell type, and then you can check some more detailed, for example, lung-specific cells and their canonical markers just very quickly in this view. So let's say we choose epithelial cells and the alveolar epithelial cells, and then maybe the type 1 cells. And then very quickly, you see the biggest like canonical markers, so gene markers of this cell type coming from this specific organ, tissue, and then specific cells. Then you can just switch between the organisms that you're interested in. So maybe try one other, maybe digestive system here from the intestine. You can also see the different tissues that every organ, like every possible available tissue for different organs here. Let's go back. And then for the, again, cell types, you just need to check which ones would you like to see. Again, maybe go to epithelial cells, and then maybe goblet cells. And then in few seconds, you can just reach the canonical markers of those specific cell types. So this is one type of search that you can do in SynEcoSys very quickly in just few seconds. Let's go back to the home page. Maybe next time you would like to search by genes or cell or studies. Let's go some of the cells that I already searched for. So just one as one of the examples, I searched for the B cells. So you can just search also for much deeper cell types for, so now I just typed like a major, bigger cell type, but you can type or search for very like more in-depth cell types, which are more like subtypes of bigger cell types, and see the details of those cell types in this page. So first we see the basic information about the cells, the ontology name, the synonyms, the definitions. So for some specific cell types, there are descriptions of also coming from like protein information and the description of the protein, etc. So you will see like a basic summary information about the cell type first. And then you see the canonical markers of that cell type. So yeah, B cells, it's a bit general, but still you see like the first for each different organism, you see the gene markers of those cell type and the evidence numbers and other expression in target cell percentages around here. Then the next one is actually very helpful, the cell ontology. So from left to right, you can follow the direction of differentiation. So from left is the bigger group, and to the right, you just go in depth to see how this cell type is actually differentiating. So obviously for other cell types, for more in-depth cell types, you will see like a bigger, like a tree here. So it's also very useful. Then we have some database-wide statistics. So like the availability of this specific cell type in different organs and tissue. And then if you check these numbers, you can see the numbers of how many diseased or normal samples this cell type is actually found. For example, like in our database, so in many normal tissue, B cells are found, but also head and neck cancer, and also this like one pharynx cancer, etc. So you see like a big database-wide statistics between different like organs and tissues around here. Then, we have some SynEcoSys core datasets. I think I will go into detail about these just a bit later. And then this search will finally return you all of the publications or like data sets that these cell types are actually mentioned. So this is like the whole number of data sets, and now you see this many this many data sets actually has B cells in them. And here you see like a nice summary of each publication at a glance. But if you like to see the details of each paper, you can just view or then save for later to take a look in a later time. So another thing you can do, so maybe you would like to compare different studies or like data sets in the SynEcoSys. Let's say you just click on these like different publication data sets. So these are all different data sets coming from different publications. And if you click on the browse and compare, obviously now I just randomly selected some data sets and studies, but then you can actually compare those data sets and maybe some cell types, like let's say maybe melanocyte cancer cells. Maybe you can just select some of the cell types from each data set and then calculate the cell type distribution levels. Okay, maybe this one is not a good option because it had only coming from one data set. Let's maybe choose another cell type. Let's see, like maybe effector T cells. So you can very easily compare like cell type distribution coming from different data sets. And if you actually done your research before and like find some more meaningful comparable data set with each other, you will see some results very quickly. I mean, some basic comparison results in just few seconds. Okay, let's go back to homepage. Then you can also search per study. So if you search the word cancer in the paper titles of each study, you will end up seeing this page. So I just browse for cancer, and it looks for the publications which have cancer word in the paper title. So we have this many, almost like 200 data sets in the database, and you see some like statistical of sample numbers and the subtypes coming from all of these data sets. So for cancer, yeah, we have mostly lung cancer data sets and then intestinal cancer, etc. But then you have like also the disease subtypes here. So they're like the diseases are actually very like refined in detail. So you can also search for disease subtypes here, and you see the distribution of those diseases around here. And then yeah, there are some more statistical like numbers where they research areas and different sample types. Yeah, and then yeah, when you search for studies here, this like summary shows you like some very meaningful to the point information. So each coming up study has a SynEcoSys ID with a dataset ID, so that you can use this for also search reasons or for your analysis in the later in CeleLens. So if you like to use some SynEcoSys datasets to integrate with your own data, you will need this ID to enter so that this dataset can be extracted from the database itself. Then you have some summary information, how many cells are there, how many samples are in this dataset, coming from which organism, if sorted, if it's treated, etc. So here we actually have also this nice, very detailed search filtering options. So these filtering options are actually very detailed in clinical data. So different clinical data levels can be filtered around here. starting from model views or species, organs, tissue, disease subtypes also, so you can filter for different disease subtypes. What else? Different stages of different diseases, different type of treatments, post, pre, naive, and different, maybe, which is most important here, the drug details. So you can search for different drugs which are used maybe in combination or just by itself. So you can filter for different levels of clinical data information while you search for studies in SynEcoSys database. So there are more options coming from clinical metadata, the ethnicity, age. There are like different levels that you can filter for. Then I think I will just briefly show you some core data sets. So SynEcoSys has some core data sets which include different disease data sets all together. So let's go to one of them. So this blood cancer. This has, like, you see, all of these data sets are actually integrated in this core data set. So you see the summary around here. They're all blood cancer from human. So it has this many cells coming from like 448 samples from different organ tissues. So you see the summary information of the integrated data on top. And then you see the study details around here. So all of these studies are integrated and also analyzed so that to find like a common ground for different organs and diseases just before for your convenience. Not for you to do it by yourself again, but there are some like the core data sets that they're actually already analyzed for you. And all integrated together. You can also use them for comparison of different mutations, different diseases, and etc. Okay, so I'm back in home page. So we also have some data statistics. Whenever there are new like data sets added, you can check them in the update log, and these numbers will they always increase by time. So it's like ever expanding database. So finally, here I want to show some of the data sets that Regina already showed in the presentation. So I went to my accounts, my own saved publications, and she actually showed this cancer blueprint, this colon cancer data. So I already saved that, and now we can just check some details in the SynEcoSys database and what you can actually see. So here we first see like a very short, like a nice summary of the publication with the ID, again with the same metadata information, the organ, tissue, mutation, the sample types, and some publication information here, the platform, journal, anything that you need to see at the first view. And then you have some research background. I mean, the summary of the research, some experimental design, and the results and significance. So very to the point information about the study itself. And then for all these data sets, the data analysis is actually the basics of the data analysis, it's already done. And the annotation of the cell types, they're actually standardized according to SynEcoSys annotation. So here you directly see the dimension reduction plots of the cells coming from this data set. And the annotation is already done for you, and it's the standardized annotation. Also having many different cell subtypes like CD4 plus naive T cells, effector T cells, etc. So this detailed analysis or annotation, at least, it's already done. And here, if you like to check like some height or see some cells, you can do it on top of the UMAP plots. Also, another important thing is just the sample groups. So here, all of the sample groups are actually, so this information, this clinical sample group information, it's already recorded also in the analysis, so that you can use these many different clinical information level groups to view your data. So for data visualization purposes, this clinical information coming from the publication, they're already recorded here so that you can use it for visualization. So here we have, I think, different maybe sample types. Yeah, paratumor, tumor. So you can, if you like to compare different like sample types, you can also do it from the UMAPs. Let's go here. So maybe sample type. So any figure can be shown in different subsets, in different sample groups here. So here it's the same information, they're the same cells, but they're labeled according to sample grouping here, not for cell type, etc. So there are like many different aspects that you can group or label your cells on top of the UMAPs. So I just want to briefly show some other analysis. So here, I think on the in the presentation, Regina show us two genes. I think I want to show them also all together. It was the KL6. Was it something else?
KLK.
Let me
6.
check. KLB, was it KLB?
KLK.
KLK. Yeah, I think
6
so.
and
Okay,
10.
I think it was KLK 10 and KLK 6. So you can just either upload your own genes list or type the gene IDs so that you want to see the expression levels all together, or also the co-expression levels around here. So it takes a few seconds. So you can, you can see the gene expression levels of the selected genes very quickly in just 5 seconds altogether. And here, these are actually corresponding to cancer cell types. If we check it here around per cell type. So yeah, those cells are actually annotated as cancer cells. So you can see it here. And here you see like their expressions are very similar or comparable on top of the UMAP. And you can also see you can compare by expression in different sample IDs. In different subjects, you can see the expression levels in different views, either heat map, dot plot, biome plot. So there are many options of visualizations. Also, you can compare by a sample group. If you select different groups of cells like tumor, paratumor, so you can do comparisons of different groups also very quickly in a few seconds. So you can check the cell quality metrics, so you can check differential expression, but also you can submit some extra analysis, some more advanced analysis like cell interaction, trajectory, CNV, and differential expression comparison. So these ones are a bit more advanced, and you need to submit your analysis and then wait for the result for a little bit. But for some most basic single cell analysis steps, they're already done for you, and it's already included here so that you can directly view them. Here you see the top 3 cell markers of each cell type in a heat map version, heat map view. And then this is like a bigger heat map with different cell types and different cell markers. Okay, I think this is a bit summary of the whole database. I just wanted to show you some details. But from there, so SynEcoSys database is actually like includes our analysis, CeleLens analysis platform, and you can directly switch it from here, from this data analysis portal view. So you can just go to CeleLens Cloud part, and then you get started with your own account. So in my account, I already have some already uploaded files and already done analysis. But this is the first view of the cloud. So it's actually a similar view as SynEcoSys. And here you see some of the uploaded files. And yeah, you can import your own data. So it's not platform dependent. As long as you have compatible files coming from many different platforms, you can upload your data. And then once they're uploaded, you can start or initiate your analysis. And you can also integrate your own local data with the SynEcoSys data sets with their dataset IDs. So once you know the dataset IDs, you can just integrate the SynEcoSys datasets with your own data. And here you just need to change the parameters for the annotations and cell filtering and some other options. And if you like to integrate different studies, it's nice to have some more advanced quality adjustments like ambient RNA removal, doublet removal, and batch effect removal. They're all here. So if you like to integrate many different data sets, you can just use these quality control steps to ensure that your data is actually comparable. At the end, you just submit your analysis and wait for the results. And the results, they're actually very similar to the ones that I actually showed you in the database. So at the end, you can just view all of the results and download your figures and data sets coming from either your own analysis or from SynEcoSys. I think my time is a bit up. So if you have any other questions, I think we can answer them during the Q&A session. Thank you.
We already have one question in the Q&A. Does the database also have multi-omic data such as TCR, BCR, and ATAC-seq?
Maybe I can answer. Okay, so the database is mainly focused on single-cell RNA and nuclei datasets for now, but we have some TCR, BCR, and site-seq datasets already available, but their numbers are like a bit low compared to the single cell and single nuclei. But depending on the demand and the requests from our customers, from our users, I think since this is like a ever-expanding database, there will be added some more multi-omics datasets in the future.
Yeah, thanks. We have two more questions coming up. So, are there any size constraints for uploading data for analysis?
Okay, so I think I can also explain this part. So normally for data analysis, you either generally upload per sample gene count matrices, and generally they're not too big, but each account, SynEcoSys or CeleLens accounts, they come with a storage limit. So as long as you don't cross that limit, it should be fine. And those per sample gene count matrices, they shouldn't be like too big. But depending on your data size, there are like different packages that we offer for CeleLens. So if you think you have very big cell numbered projects, the data storage limit can be increased.
I'm actually interested to know more about the analysis functions in CeleLens. What are the types of analysis that can be done, and is there a user guide?
Okay, I'm all again taking over. So the CeleLens focused on mostly single-cell RNA data analysis, and it can do any basic single-cell data analysis workflow. On top, we also offer some like advanced steps like trajectory inference, trajectory analysis, cell-cell communication analysis, CNV analysis, or the comparison of differential expressed genes, also pathway analysis and pathway and gene enrichment analysis, and transcription factor regulation analysis. So they're both basic functions, and there's some more advanced analysis already included in the platform.
What about data security? What happens to my dataset when I upload on CeleLens?
Yeah, maybe I take that one now. It's a very good question and a very important topic. We can assure that the data will not leave the software. The data is imported and processed and analyzed, but is not collected by SynEcoSys. And the rules, of course, the GDPR rules and other compliance rules are followed, and the data is not transferred to any other location. So there is no, let's say, way the data could be used by someone else or seen by someone else. And as soon as you would end an analysis, you can also delete all your data. And when the license is terminated, everything is erased. So there's nothing someone else could do with your data that you have uploaded.
Yeah, thanks, Regina. We are still very good with time, so I will go ahead with 2 more questions. Can I get more information on product options, licenses, and pricing? Also very important.
Yeah, sure. And again, my questions, sure. We will answer all those questions one by one, depending on the needs of the person requesting. Please leave us a note in the chat that you want to be contacted, or send us an email to our email address. It's bd-team@singeron.bio, and I'm sure it will be shown or mentioned later on. And then we will share all information needed.
Yeah, thanks. So for everyone, I also wrote the email address on the chat. You can get in touch at bd-team@singeron.bio for more questions. And the very last question that I have here is it possible to try out the software?
Yeah, sure. You should try out if you're interested now. So that's the idea, that we ask you to try it out. And if you're not quite sure if it serves your need and you want to check out, we offer you, as a participant of this meeting here today, a free trial for 1 week. Again, drop us a line, send us an email, and you will get the possibility to try the CeleLens and SynEcoSys for 1 week for free, and then you can decide for further action.
Thank you so much for joining, and see you on the next one.
See you.